Okay, hi everyone. Sorry, I got off to kind of a slow start there. I'm still trying to figure out my new computer setup here. All right, so let's continue talking about um, metastases, right, and the different types of factors and changes that take place that both influence the spread of cancer cells from a primary tumor site, and then also understanding the different challenges that these cancer cells face when they are spreading throughout our body. So we're going to talk about the motility of cancer cells and also this important event of epithelial to mesenchymal transition in more depth. Okay. So let's remember some key concepts here. Cellular transformation involves the buildup over time of mutations over many generations of cell division. Right, so we said that for most of the semester, we've been thinking about that as just sort of like a monologue, right, of this transforming cell kind of undergoing its own sort of private sort of set of issues, right, changing and things taking place over many years of different mutations building up. That's why the time it takes for a primary tumor to actually build up to a substantial cell mass takes many years of one transforming cell building up in numbers and then being able to have different populations, subpopulations of those transformed cells, each beginning to get different sets of mutations, right? So the individual cell that gives rise to the tumor over time becomes a heterogeneous clonal expansion of different pockets of cancer cells within the same tumor. We also learned that in order for that tumor, once it reaches a certain size, to be able to overcome different limitations imposed upon it by its size, right, including the ability to break out of its in situ physical boundaries of things like a basement membrane, that requires the help of a manipulated stroma surrounding it that's supporting the change. And that's why the last few lectures we've been getting to appreciate more how it's not so much a monologue of this one transformed cell, right? It's actually now a dialogue having to do with how that transformed cell is now changing the way it is supported by the surrounding tissue. So if we continue with our kind of analogy here of the rebellious teenager, so at a certain point, right, the confused parents who are manipulated and don't know what to do up until now, right, the teenager still has really depended upon the parents for support, right? But maybe at a certain point, at a certain breaking point, right, things just get so bad and maybe one of the parents, I'll just assume it's the dad, right? Because I don't want to say it's the mom. Maybe at a certain point, the dad just has had it, right? And says, that's it. You're no son of mine, get out of here or whatever, right? And so the teenager runs away from home. But is he really prepared for what's ahead? Right? So that's what we're going to talk about today, are the things that a tumor cell has to deal with once it's been able to break free from its in situ environment and now is sort of out there, right, in the bigger world of the tissues of the body. When we look at different examples of metastases, I think you're all aware that there's tremendous variation in the types of morphologies, the types of phenotypes of metastatic cancer cells. And much of that, as we already examined, has to do with the fact that the cell that has broken away from the original primary tumor, it has spent all of its time accustomed to that particular environment where it is used to living, right? Because the original cell that it came from was designed to live in that environment. But once you have cells that end up being in different types of tissues away from the original sort of type of stroma and supporting tissue that cell originally was familiar with, right, things can take on very crazy sort of changes. So here are just some examples of 3 different metastatic lesions representing different cells that are now starting a secondary tumor in some sort of distant metastatic site in the body. So here the first one is pancreatic cancer. So these are pancreatic cancer cells that have taken up residence inside of a lymph node, right? So many cancer cells as part of metastases. We already learned that the lymphatic system can play a role in this because the lymph system is constantly in charge of draining things from our tissues, ostensibly to be able to clean things up, right? And get the water back and reintroduce the water back into our circulatory system. But that's why the lymph system is a very readily available source of or method of cancer cells escaping from a primary tumor, right, and spreading throughout the body. So here's pancreatic cancer cells that have made their way into a lymph node. Here's breast cancer that's in a lymph node, right? Very different. Here is metastatic lesions in bone marrow. So very different appearances because these are very different types of scenarios. So a primary tumor has to have about 10 to the 10 cells before it can be detected, right? That's 10 to the 10 cells before a patient can actually be, let's say, at a checkup or some sort of clinical visit, and they can actually see that there is the formation of a tumor. The important thing is primary tumors only account for about 10% of cancer mortality. The vast majority of mortality associated with cancer comes from metastatic disease. That is, after the cancer has spread from the original tumor and is now causing these secondary distant lesions in the body. So that's really what comprises 90% of cancer deaths, right, is metastatic disease. And there's high variability in metastatic tumors, depending again on the primary tumor type. So we are revisiting again this theme that we've had throughout the semester, that if you can know what the original cell and tissue type is that gives rise to a tumor, you have a head start in being able to understand what types of intracellular pathways might be the most targetable, right, in a particular tumor to have more efficient, timely therapies. So the process of metastases takes place over many steps. We can think of it as 7 steps that we'll call the invasion metastases cascade, right? A series of events that happens one after another. So today we'll kind of take this apart and take a closer look. So before step 1, you have the primary tumor formation. So you have these blue carcinoma cells that have begun to kind of learn how to do the angiogenic switch, right? That means they've now stumbled upon mutations that are now attracting vasculature to grow and continue feeding these cancer cells with oxygen and nutrients that they desperately need. Remember, if the tumor gets too big and it hasn't yet stumbled upon these angiogenic mutations, right, you're going to have a lot of necrotic tissue forming as the cells in the middle of the tumor are cut off from oxygen and just die away, right? Also, because the tumor itself is going to be contained in situ because of being in the basement membrane, right? So part of the angiogenic switch is getting the vasculature to grow and also being able to trigger that wound healing response that's going to break down the basement membrane, suddenly not only releasing the physical limitation that this tumor is facing, right, but also releasing all of those mitogenic types of signals that then were kept in the basement membrane. Suddenly, these cancer cells are doused with them, right, and they begin to grow really fast, and that also encourages the blood supply to come in. And so here you can see, right, you begin to have this primary tumor cell here, right, beginning to migrate out, right? So step 1 is localized invasion. Right? So localized invasion has to do with what we discussed already. So some of these cancer cells may have stumbled upon mutations that wake up the expression of platelet-derived growth factor, or PDGF. as well as VEGF, vascular endothelial growth factors, right? And these will recruit things like immune cells in the stroma. So macrophages will come. And the macrophages think, oh, we're dealing with a wound here. So the macrophages release things like those enzymes, proteases, like matrix metalloprotease 2 and 9, that remodels the extracellular matrix, right? Because during a normal wound response, you're trying to get rid of damaged tissue so that fresh tissue can grow in its place, right? In this case, right, this leads to the dissolving of the basement membrane, the release of growth factors that have been contained in the extracellular matrix, right? And then over time, you begin to build the stroma, getting increasingly confused and reactive, right? So the stroma is reacting to this confusing set of signals that is being sent out by the tumor. And this encourages further invasion. Right? So some of these things that happen then during step 1 to encourage invasion is you have changes in the expression of proteins that normally keep cells where they're supposed to be physically. So we've studied a number of different cell surface proteins that are involved in cells detecting their surrounding extracellular matrix to let them know that they are where they're supposed to be, so they should be happy and just continue doing their normal differentiated job, right? Some of these are cell-to-cell communication proteins, like the cadherins. So here we have a melanocyte in our skin, right? And it is normally helping out the keratinocytes. So remember, one of the important functions of a melanocyte is to produce melanin if these keratinocytes need the melanin because they are experiencing genomic DNA damage from UV light. Right? Remember, the keratinocytes themselves, if they get damaged, right, they'll just undergo apoptosis if they cannot fix their own problem. Right? If the problem isn't that bad, but there still is some damage, it's not a big deal because these keratinocytes aren't supposed to divide maybe more than 2 or 3 or 4 times, and eventually they're just going to get lost anyhow, right? But the melanocyte is not supposed to go away. So here you have the melanocyte joined up with the keratinocytes so that it does its normal functions. But what happens if the melanocyte gets damaged, right, by UV radiation and it doesn't properly undergo apoptosis? Right? One of the things that might happen as it becomes a melanoma cell is because its mutations are causing it to get messed up, it may actually switch between the normal E cadherin, shown here, right, the blue ones, that normally allow it to preferentially associate with epithelial cells. It may end up turning off the expression of E cadherin and instead start expressing N cadherins. I mean, after all of these genes, these different variations of cadherins are all part of the genome, right? It's just normally the melanocyte would have the N cadherin genes maybe shut down, maybe through gene regulation, or even things like hypermethylation of the promoter, right? But in this case, what's happening is that now there are mutations that have switched on the N cadherin. The N cadherin is the type of cadherin that's used by mesenchymal cells, the stromal cells, right, to detect that they are happily part of the stroma. So this melanoma cell then begins to no longer care about hanging out with the epithelial cells. It begins to care about hanging out with the stromal cells. For example, fibroblasts that are in the stroma and endothelial cells, right, that make up the walls of things like our capillaries, right? So you can already imagine this encourages the melanoma cell to start wanting to migrate through the mesenchyma. Okay, once those cells have left the original boundaries of the basement membrane, for example, and are now happy spreading through the stroma, right? You can imagine now these cells spreading away from the primary tumor, the in situ site, right? Very soon, they're going to encounter something like a capillary. Remember how extensive the capillary network is through all of our tissues, right? The cells in our body are no more than maybe like 0.2 millimeters away from the closest capillary, right? So as these cells are spreading through the stroma, yeah, right, they're going to be coming up against capillaries, right? And because of their newly found liking of associating with things like endothelial cells, they are going to now enter the capillary circulation. So this is step 2, intravasation, right? That's the term given to when these tumor cells make their way into the blood. So in order for this to happen, you have the sort of perfect storm of different cells working together, being tricked or fooled into doing their normal functions that are normally supposed to be beneficial, right? Helping us during wound healing, for example. But in this case, they end up actually supporting and helping the tumor cells. So things like the carcinoma cells themselves, sending out all kinds of weird messages because of their mutations, macrophages that are supposed to come and kind of help out, they get confused, right? And so they continue to sort of dissolve away the extracellular matrix, making it even easier for the carcinoma cells to spread. And the endothelial cells, right, the walls of the capillary, they get confused as well, right? When these tumor cells press up against them, right, these endothelial cells kind of get out of the way and they let the tumor cells in. Also, once in the blood, the tumor cells are attracting lots of platelets, again, because of the perceived wound healing response. Right? One thing to keep in mind is that once these tumor cells have made it into the blood, right, this is actually only the start of a whole new set of challenges, because the blood is a very hostile environment for cells that are not designed to be normally present in the blood. All right, so let's talk about these things. So here you have some slides showing this kind of perfect storm of 3 different types of cells working together. So these are carcinoma cells that are stained pink, looking for this protein called MINA, right? So MINA is a protein that regulates actin. So actin, of course, is this important cytoskeletal protein that's involved in motility of cells and also adhesion of cells. And so you oftentimes see the overexpression of this MINA protein associated with invasive cancer cells, right? So these, in other words, are cells that, as part of the cadre of random mutations that they stumbled upon, here they've learned how to upregulate MINA, right? And this is now helping them become more mobile. In these circles and dotted line regions, you can actually see 3 different cell types coming together. You have the endothelial cells that are stained sort of bluish here. So this would represent the capillaries, right? So these 3 sort of spots here that are pointing to by these lines represent capillaries in cross-section. So you'd have endothelial cells there. You have the macrophages, right, that are also here sort of working to dissolve the extracellular matrix, making it easier for these carcinoma cells to make their way through. And of course, you have the carcinoma cells themselves. Right. So in these regions, you can see that in these circled ones on the right, you can see that same thing, evidence of macrophages, the carcinoma cells, and the endothelial cells of the capillaries working together. One thing to keep in mind is that capillaries are very limited spaces. Right? Remember, the capillaries are the teeny tiniest arteries that are spreading through our tissues to supply all of our cells nutrients and oxygen and take carbon dioxide, etc. A typical capillary diameter is about 10 micrometers. To put this in perspective, a red blood cell that's flowing through that capillary is itself about 7 micrometers in diameter. That is why, right, red blood cells that are flowing through the capillaries are almost doing so in single file. Right? So in this picture here, you can actually see an illustration of a capillary with red blood cells, white blood cells in it. So the endothelial cells, or the wall of the capillary, is in E, a red blood cell here, right? It's being kind of squeezed and squished as it floats through. But remember, a red blood cell is a biconcave disc, right? And so here it is sort of viewed on its side. Right? And then a white blood cell, larger white blood cell, is in W. So I can actually illustrate it this way to give you an appreciation, right? So the width of the capillary, right, is not that much bigger than one of these cells itself. A cancer cell, on the other hand, is pretty big compared to a white blood cell or a red blood cell. a cancer cell might be on the order of 20 micrometers or larger, right? So it's actually going to be really crammed in this capillary as it's being sort of shoved along by the flow of the blood, right? So consider this, that itself is physically rough for the cancer cell that has squeezed its way into the capillary. So the circulation in the blood is a very rough environment because these carcinoma cells that maybe were designed to be, let's say, glandular cells or something like that, right? They were never meant to be designed to flow through the blood. In addition, once they're in the blood, oftentimes these cancer cells, right, are recruiting things like platelets, and they end up becoming these masses of cancer cells, right, coated in platelets. If you look at this here, that's what we're seeing here. These are cancer cells in circulation, and they are have with them these little bits of things. You know, I think these little green things are probably actually platelets, right? And so this creates this mass of cells. So on the one hand, the platelets kind of protect the cancer cell, but this whole thing is being just shoved through the capillary, right? Pushed by the blood flow. So it's almost like, you know, if you've ever gone whitewater rafting, right? It is a really rough place for a cell that's not designed to handle it. So as a result, what happens? A lot of cancer cells just get destroyed in the blood. They just literally get torn apart. Okay, the next step, right, is that these cells, you know, as they're getting shredded apart, you know, not all of them will get shredded apart. They make their way through the circulation. Eventually, they make their way to the blood and they get pumped, right? They get pumped into organs like the lungs or bone marrow or the brain. So as these cells are flowing through our circulation, we call them circulating tumor cells, or CTCs. But these CTCs, within just a few minutes, are going to get lodged. They're going to get stuck in capillaries in different organs, right? Because even though they made their way to the main blood supply and now they're getting pumped out by the heart, once again, they're going to go through a series of arteries that are branching and getting smaller and smaller and smaller, right? And by chance, maybe some of these cells ended up in the lungs or up in the brain or other organs. But at some point, these cells that are getting shoved along, they're going to find their way and get jammed in the capillaries again. When they oftentimes get jammed, they can actually break apart into little pieces. So many cells will get broken up and die, right? But cancer cells oftentimes can lose a tremendous amount of their cytoplasm and really become nothing much more than a bloated nucleus with a little ring of cytoplasm around them, right? And that causes them to maybe dislodge and come loose, right? And continue on being spread around. But that's why many metastases do end up in the lungs, because the lungs are the first place that circulating tumor cells get pumped to by the heart, right? Because remember, the heart pumps the deoxygenated blood up to the lungs through our pulmonary veins and arteries, right? And before returning to the heart, and then the pump getting the heart pumping that blood out to the rest of the body. So many cancer cells will just get stuck in the lungs. So that's why you oftentimes have metastases take place there, right? But for those other circulating tumor cells that make their way to other organs, right, they sort of continue their journey. Right? So here we have circulating tumor cells. Here's a cancer cell. These little blue things are the platelets that it's sort of attracting. It's making its way through a capillary here, right? And eventually it's going to get stuck, right? So let's say it gets stuck at this point here. So here you have the wall of the capillary. The green represents the endothelial cells on either side of it, right? And then the pink would sort of represent the boundary, right, of the sort of that edge of the endothelial cell. And then the gray on either side of it would be the tissue surrounding the capillary, which is oftentimes just called the parenchyma, right? It is the tissue surrounding the blood supply. Right? So as the cancer cell gets stuck there, it gets stuck and it begins to get coated with more and more capillaries, right? Not capillaries, sorry, platelets. as it continues to attract platelets. Right? And then it forms basically a little microscopic clot. Right? So it forms a microthrombus, right, which is a blood clot. Right? So again, this is another example of our body's physiology then being fooled into helping the cancer cell. Why? Because obviously we have mechanisms for preventing these microthrombus clots from forming in our tissues, right? So when these clots tend to form, right, cells express different types of proteins to break up these clots. And these proteins cause the platelets to disassociate, and they cause the walls of the capillary, right, to kind of give a little bit, to try to encourage this clot to be able to pass. In doing so, what happens is that this cancer cell then gains easier access to the basement membrane that separates the endothelial wall from the surrounding parenchyma. Right? So the cancer cell begins to push aside the endothelial cells, and once it gains access to the basement membrane, something interesting happens. Because remember, a lot of why this cancer cell got all sort of rowdy in the first place when it was rebelling was because of that kind of toxic home environment, right, back at the site of the primary tumor, because things had gotten really weird in terms of the dynamic between the primary tumor cells and the surrounding stromal cells, the reactive stroma, right, that kind of represented like the parents of the teenager who were just so fed up and confused, right, that the household just became intolerable and this cancer cell escaped. It was encouraged to escape. But now that it's somewhere else in the body, very far away from that original reactive stroma that was giving it all the sort of confused messages that it was using to kind of escape in the first place, it's no longer receiving those reactive stroma messages. So in many ways, the cancer cell begins to kind of chill out, right? It calms down in a way, right? And it wants to revert back to a more familiar type of environment that it's comfortable in. Right? I once read a report about runaway teenagers that said something like, a majority of runaway teenagers are found within the first 48 hours of them being reported missing. And interestingly enough, 80% of those runaways who are found very quickly, they're usually found at places like the mall or at fast food restaurants. Why? Because once the teenager has had some time to kind of cool off, right, away from fighting with his or her parents, right, away from home, they calm down and begin to seek out things that are more comforting to them, things that they are familiar with, right? Things that make them feel safe, like hanging out at their favorite mall or hanging out at the McDonald's or something like that, right? So circulating tumor cells are like this as well. Once they are far away from that kind of toxic environment of their original home, their primary tumor site, right? Once they start attaching to something like a basement membrane somewhere else, right, they begin to want to sort of resume their previous function, right, of being more epithelial. Okay. So this process then, right, of then wanting to squeeze out of the capillary because they're attracted to the basement membrane on the other side of the endothelial cells, right? We call this the opposite of intravasation. This is now extravasation, right? So they squeeze their way out and they take up residence in their new stromal home, right? Presumably a stroma that is very normal, right? That is a well-behaved stroma. And here they may begin to continue their out-of-control dividing. But usually this stroma is going to be very different than the stroma that they started off in. Right? Even though it's not crazy reactive stroma, right? It is stroma that is designed to support epithelial cells of a different type. Right? So the types of cytokine messages or signals that this stroma gives out, might not be a very good match for what these little blue tumor cells are used to, right, compared to what they have back where they started off. So as a result, yeah, they'll continue dividing, but it's probably going to be kind of slow because they're not really getting the types of messages that they're used to, right? So they form a micro metastases, literally just a handful of cancer cells, right? And then they just kind of stop because they really don't know what to do beyond that, right? So here you see, here's the process of extravasation, right? So that cancer cell, it has formed a microthrombus clot covered in platelets, but enough platelets have covered it so that our body, our tissue reacts by saying, uh-oh, there's a clot. Let's do some things to break up these platelets here, right? And so the platelets get broken up and sent on their way, and the capillary wall becomes a little bit more forgiving, right? To help sort of pass this, this clot. But as a result, that allows this cancer cell now to find its way and attach to the basement membrane, right? So it's sort of happy again, because this is sort of like, oh, you know, a basement membrane. I've missed having a basement membrane. It multiplies a little bit, but then it just kind of stops, right? Because the stroma surrounding it is not the type that it really knows sort of how to kind of happily live in. Right? So here are some kind of remarkable images here. So here is an arteriole, so thin artery, and then each of these, of course, represents the branching off of that into teeny tiny capillaries. And you can see here, they have stained cancer cells, these circulating tumor cells, CTCs, right, in this bright green fluorescence. And you can really see, right, that these cells were flowing down this larger arteriole, but once they get down to the level of the capillaries, they are now lodged there. They're stuck, right? But given some time, they make their way. Look at this one here. You can tell it has now squeezed its way out of the capillary and is now in the surrounding tissue. So this process of a cell in the blood squeezing its way out of the capillary into surrounding tissue, this is what we call diapedesis. This is normally what immune cells do. So for example, monocytes that circulate in our blood, when they arrive at the site of a tissue infection, they will undergo diapedesis to squeeze their way out into the surrounding tissue and become macrophages, right? Neutrophils also do this as well. So cells that are meant to do that, like certain immune cells, they can do diapedesis in just 1 or 2 minutes because that's what they know how to do. That's what they're designed to do. However, a circulating tumor cell, right, it doesn't know how to normally do diapedesis. It doesn't have the right pathways necessarily active in it. So it's not going to be able to do this in a couple of minutes. It's probably going to take hours or days before it squeezes out of the capillary. Right? But the important thing is, once it is in the surrounding tissues, we now no longer call them circulating tumor cells. Now they are stuck somewhere, right? So we call them now disseminated, right? They have now gone to a new location in the body where they are now fixed, right? So they become DTCs, or disseminated tumor cells, that are forming now small little micro metastases tumors. So here you have some images of micrometastases. So these are colon carcinoma cells that have metastasized into bone marrow. So the blue is the bone marrow, and here you have, right, 3 colon cancer cells that are now disseminated. They are now fixed in the bone marrow here, right? And they are kind of dividing, but they're sort of like, what is this place, right? I don't know what this place is. And so they don't really go very far in terms of dividing. Here's breast metastases into bone, right? So in the bone, this breast metastatic cell maybe made its way in here, and then underwent diapedesis to squeeze its way out of the capillary. But once it's in the bone, it's kind of like, you know, okay, I don't know what this is all about. So maybe it only divides a few times, and you get a micro tumor, right? That is maybe like 12 cells or something, right? Okay. So the important thing to keep in mind is that these DTC microtumors can lay dormant in metastatic sites all over the body indefinitely, right? So again, here's an image of breast cancer cells that have metastasized to liver. So the green around it, this olive green, is liver cells, and these bright fluorescing things are the micrometastases. So think about this. If you have a primary tumor that has undergone angiogenesis, has done the angiogenic switch, right? So it's learned how to get the blood supply it needs. It's also now triggered the wound healing response, so that now you have cells breaking free of the basement membrane in the original primary site, making their way through the stroma, right? We said that the stroma is filled with capillaries running this way and that, just a few micrometers away. So just think, right? Once that basement membrane is dissolved, right, and all those cancer cells are beginning to spread away and encountering capillaries, right, you could have thousands upon thousands of cells being shed by that primary tumor every day, right, into the circulation. And these circulating tumor cells are now going all through the circulation, getting stuck in different sites of the body. That means by the time a breast cancer patient presents in the clinic, right, with a sizable tumor, right, in the breast, right, that patient may already have thousands of little micrometastases tumors spread out in all the different tissues. But these thousands of different metastatic microtumors, they're in places that those cancer cells don't really know what to do next, right? Because it's very foreign stroma. One problem with this, though, is that because these micrometastases that are spread out through the tissue, they're too small to be detected, right? Because they may only be a handful of cells, and they're not really actively dividing. Right? So as a result, when that patient gets different types of chemotherapy treatments that are meant to target the primary tumor and try to get it to shrink, right? That primary tumor is still going nuts, right? It's still rapidly dividing. So many drugs are meant to target cells that are rapidly dividing, right? So the primary tumor cells might get hit very hard by that chemotherapy, by that particular drug, and the primary tumor may regress, right, and shrink. But the thousands of micrometastases, metastatic lesions that aren't rapidly dividing, they may not actually be affected by the drug, right? Because they are not rapidly dividing. So this brings up the seventh stage in this invasion metastases cascade. Right? I mean, it's one thing to have the body scattered or littered with thousands of little, micrometastatic lesions that are like a handful of cells, because honestly, even though that sounds really scary, if those micrometastatic lesions don't actually start dividing again and they just stay there as a little handful of cells, eventually some of them will just go away, right? And they're not really disrupting the function of the new tissue that they've ended up in, right? So of course, the real problem happens when some of those micrometastatic lesions get happy with their new environment, right? Or by chance, this is the other thing to think about, because they kind of were randomly scattered throughout the body in different types of stromal environments, if by chance certain micrometastases start off in another stromal environment that's similar enough to what they're used to, or, right, may ironically be even better than what they're used to, then you have problems, right? Because then those micrometastases will start to proliferate. So that's step 7 then, is the colonization and formation of a macro metastases, right, from one of these micrometastases somewhere else, and thereby completing this awful kind of cascade where you now start having a secondary tumor growing now at the rate of the original primary tumor, but somewhere else in a different tissue in the body. Right? So some bullet points here, right? The DTCs must be happy with the new tissue environment in order to form a macro metastases, a new large tumor. Right? The vast majority of DTCs don't proliferate easily in their new tissue home. And consider this, in order for them to sort of continue where they left off and become another large tumor, they actually have to revert back from being more mesenchymal to being more epithelial. Right? So this brings up a very important concept in cancer, which is the seed and soil model of metastases. Right? So this model, this sort of paradigm or analogy, has been around for several decades, but it still holds true today. You can think of a circulating tumor cell as being like a seed of a flower. Right? And if you have a handful of these seeds, each one of these seeds has the potential to become a new flowering plant. Right? So imagine that if you were each given a handful of these seeds and you wandered around campus, and at random locations, you just scattered a few of these seeds. So you scatter some, at the base of a tree somewhere, you scatter some on the lawn, maybe you scatter some in a parking lot, or you scatter some, by the side of a building, right? And then you come back a few months later, you would not be surprised if some of the places where you scattered the seeds, right, you have now all these nice new wildflowers growing. While at the same time, you wouldn't be surprised if you go visit the parking lot, and you see the seeds that you scattered onto the pavement, there's nothing, right? Those poor seeds were scattered around randomly, just in the same way that a circulating tumor cell will be scattered around randomly, getting stuck wherever they get stuck in your body in the capillary system to become a disseminated tumor cell. Right? But most DTCs won't flourish in their new tissues because the new environment is simply not suited for these different cell types. So therefore, the emergence of a macrometastases really does represent a rare subset of DTCs. So this seed and soil, I think in most discussions of cancer, this model is brought up. And I think it is a nice model because I think intuitively it makes a lot of sense. And it makes you realize, right, that even though a patient may have thousands of micrometastases spread around, right, that really the ones that you care about are the ones that ended up in places where the new environment is conducive and supportive to those cells really taking off and forming a new tumor. So consider this, right? So the primary tumor, by the time it's gotten very large in its sort of in situ site where it started off, it represents many subpopulations, right, of different tumor cells with different sets of mutations. Hey, remember, I want you guys to always remember that somewhere in there, let's imagine this pink one here was the original set of mutations that maybe started this whole tumor, right? But over time, many years, right, this pink cell had divided, so it started forming a population of just pink cells, right? But as you got more and more of them, and remember, it took a long time because along the way, most of them are dying off because they're doing the right thing and undergoing apoptosis, right? But the longer you have these cells sticking around, the more chance for more mutations. So over time, these pink cells begin to diverge as different pockets of them begin to randomly pick up their own set of different mutations, right? So that's why you end up with a large tumor. That's really kind of messy inside. You have red ones, blue ones, green ones, orange ones, gray ones, right? So that's the starting point of this large primary tumor. And then some of these cells undergo EMT, right? So the large tumor then does the angiogenic switch. It triggers the wound healing response. And so now these different cells of different subpopulations are now making their way through the stroma, right? Maybe some of them don't, because maybe some of them, for example, the navy blue ones here or the green ones, right? They don't have the right set of mutations to take this any further, right? They don't end up spreading into the surrounding tissue, right? Because they don't have those mutations that allow them to do so. But these cells do. So these get into the blood, right? And they go somewhere around the body and they get stuck and they form micrometastases. But let's say that out of all of these, only the blue ones by chance ended up being somewhere where the new stroma environment is very conducive to these cells really taking off, right? So that means they need to revert back, right? Mesenchymal to epithelial transition, right, is almost like them going back to their original epithelial form, so that when they start growing now, the new tumor, right, sort of takes up where these blue cells left off in the original tumor. But now the new tumor starts off just being all these blue cells, right? Because that was the right combination of the right kind of cell ending up in the right spot somewhere else in the body. So this slide is a nice way of visualizing these processes. In other words, the reversibility of EMT, right, which is MET. So here we have the primary tumor, and in this primary tumor histology slide, in red is cytokeratin, which is a marker for cells that are more epithelial-like, right? So in the primary tumor, most of these cells are still very epithelial-like. The green, I think you guys know what this green is. That's the basement membrane, right? So this is staining for lamin, protein in the basement membrane. So you can see in the primary tumor, right, they are still confined by this basement membrane. And in the black, which is not stained, would be the stromal cells that are in the spaces surrounding these primary tumor populations in here. So when they undergo EMT, right, they become invasive. So you can see the basement membrane has broken down, and you can see the cells as they spread, right, they kind of lose the red staining. They become more black, right, because they are turning more into stromal-like or mesenchymal-like cells that allow them to be happy, spreading away from the original site, wandering through the surrounding stroma. And you better believe, right, even though you can't see it in this picture, there are many, many capillaries floating through here as well, right? So many of these cells are coming into contact with the capillaries. Once these cells circulate and are disseminated, and if some of them end up in places that are conducive to them starting a new big tumor, right? Because they'll end up somewhere and form micrometastases. But some of these will be the seeds that have been scattered onto the parking lot. They're not going to grow. Some of them are going to be the seeds that you scattered into an existing flower bed already on campus, right? So those are the right conditions for those seeds to take off. So those will undergo MET to convert back into being more epithelial, right? And you can see then this cancer has now metastasized to the liver, for example, right? And when you take a cross section of that tumor, it looks very similar to the starting primary tumor, right? So those relatively rare micrometastases that have lucked out by growing in good soil will resume their epithelial-like growth activities, right? And the evidence of this is the fact that you see a reestablishment of the basement membrane. And when I say relatively rare, I think you understand relatively rare still is problematic if you're talking about every day hundreds of thousands of metastatic cells leaving the primary tumor and circulating around in the body, right? Okay, last slide for today is just, this is just, again, a different way of summarizing what we've been talking about, but in a more cartoony form, right? So the versatility of EMT. So here you have the in situ carcinoma that's being limited by the basement membrane and the normal stroma. As the tumor gets bigger and bigger, it's getting stressed out by the challenges of needing more nutrients, needing more oxygen. It stumbles upon mutations that allows it to go the angiogenic switch and also begin to attract things like the wound healing response. So you have a breakdown of the basement membrane, and you have some of these now invading into the reactive stroma, right? The stroma itself now is getting more and more confused, and unfortunately becomes that dynamic, which I said is like a toxic environment in the home, right? That unfortunately encourages these invasive cells then, right, to get into the circulation. They end up somewhere else where they leave the circulation. And if these tumor cells by chance end up somewhere, right, where they then begin to start growing, they first grow into a micrometastases, right? Most likely, it's not going to grow much bigger than that because the stroma that's down here is very different than the stroma that they're used to. But if they end up in stroma that really supports them, right, then you would have the formation of a macrometastases. All right, so let's go ahead and stop there.